All right, so we've covered carbohydrates and lipids, and now we're going to go into proteins. And I'm trying to be detailed enough and go slow enough that this is all understandable. On the other hand, there's a lot of stuff to cover, so the lectures are getting longer than I want them to be, and I'm trying to shorten them up. So I'm going to try to keep this one short. I might start breaking them up more into smaller blocks as well. So proteins, what are they used for in cells? Well, more questions, what don't they do in cells? They do pretty much everything that carries out life. I mean, it is actually carried out by proteins in some way or another. All right, so proteins are small sort of molecular machines that act as enzymes, speeding up chemical reactions. Membrane pumps. So when we talk about membranes, that says membrane, it's a little weird. They're going to be moving things across the membrane from one side to the other, creating concentration differences. Right? And it's going to be because the proteins that are going to be doing that. Specific bonding. So when one cell attaches to another cell, we said carbohydrates could be sticky and they could stick things together. But it's not specific. It's kind of like Scotch tape might stick to certain things. It's not really specific for it. But there's other things that might attach, like a piece of Velcro attaches to a certain other side of Velcro. You touch Velcro to this glass, it's not going to stick to it, but it can form a strong adhesion if it's with something else that's very specific for it. And proteins are like that. They're very specific types of bonding where there's certain things they recognize, and when they do, they attach to one another in ways that hold us together. I mean, hold our cells together, hold our whole bodies together. Okay, so they control and regulate DNA. They control and regulate really, all metabolism. Like I said, and everything that happens with the membrane. So pretty much everything that happens in cells is happening because of proteins. Some RNA does something. DNA doesn't do anything. It just has information. RNA carries out some activities as well, but it's usually with the help of proteins. So it's either proteins alone or proteins with other molecules that are carrying out all of cell's activity, which is, if the cell is the smallest unit of life, all life's activities are carried out by proteins. So they're very, very important molecules to have some idea of what they're made up of and how they are structured. So proteins, first off, are made up of subunits, which we did not have in lipids. Subunits are something that's very similar and repeats over and over again, repeat. We had fatty acids, but they didn't attach to each other. They attached to something else, like the glycerol. The subunit for a protein is called an amino acid. So going back to some structure, right, to talk about this. There's going to be a central carbon for the amino acid. We've got the acid part, which now you should have some idea from the other things we've talked about. The acid was, remember, there's this carboxyl group, like that. Remember, this thing acts as an acid right here. Now, amino, I haven't talked anything at all about that yet. So amino is going to be another type of functional group. That's what this is. It's something that gets attached to carbons, it's part of a chain, and gives them some function, usually makes them polar. Sometimes it adds charge or other properties, as well. There's a variety of them. This one is called the carboxyl, and this one here is called the amino functional group. It's NH2. One nitrogen, two hydrogens. I erased the little bit of the periodic table I had over here, but if you remember, nitrogen had has 5 electrons in that outer shell. So that means to get to 8, it needs 3 more. So it has to have 1, 2, 3 bonds. Carbon has the 4, oxygen has the 2, hydrogen has the 1. Right? So everything has some specific number of bonds, not just in this drawing, but anywhere, anytime. So this is the amino acid with the carbon in the middle. This bond here is to another hydrogen that carbon has. And then what is attached here is something we refer to as an R group. And that's what makes it variable. So there are different kinds of amino acids. what makes, say, the 20 different amino acids different? And there's more than 20 actually in cells, but there's 20 coded for in DNA. And what makes them all unique? It's who is that? What is that? Who can it be? What can it be? Well, it might just be another hydrogen. That's glycine. That's one of, they say, the 20 amino acids. So it could just be something like that. It could be something more complex. It could be a ring structure. It could have a lot of carbons and all kind of different things. It could be polar. It could be nonpolar. It could be acidic. It could be basic. So this is what makes each amino acid unique, but this is what makes each amino acid the same. So all amino acids have the amino, central, carbon, and carboxyl group or acid group. That's an amino acid. Now, that's the subunit. So we said subunits join together. So what we have then are, I'll just use this one, I'll leave it here. Take a second one. We'll use the same colors so we can follow along. Okay, so there's the amino, there's the carboxyl here, and I'm just going to put R up there. This part won't matter, like what that R is. This other part's the same. So two amino acids will then start to be joined together. Now, usually a protein is what joins them together, and this is what's going to build a protein. So it's interesting that proteins make other proteins, but that's because they do just about everything that's in the cell, including make themselves, in a sense. We're going to have the same sort of thing happen I showed you with one of the other reactions, where we get something called a condensation reaction. Remember, condensation was formation of water, so we need 2 hydrogens, 1 oxygen. So what will happen is from the amino group, 1 hydrogen will go away. From the carboxyl group, the OH part will go away. And they will come together to form that water molecule. And now, this nitrogen only has 2 bonds, it needs 3. This carbon only has 3 bonds, it needs 4. So then a new bond will form, and that bond is called a peptide bond. Okay, so that's a peptide bond between amino acids. You're not going to have to draw, say, full amino acids, but again, if you do practice it a little bit, it will be very helpful because it'll help you answer questions about amino acids. So when I ask you a question about the structure of an amino acid, if you could draw it, it's very easy for you to answer the question. If you don't know how to draw it, you might have memorized the answer, or you might have memorized some detail about it, but it'll be a little more difficult and take a little more time. And as I said, when you're doing exams, there's going to be a very set time limit, and anything that takes you more time is going to hurt you. You're going to leave questions unanswered. So the better prepared you are, the quicker you'll get through it, and the more you will get through it and answer all the questions and hopefully answer them correctly. So peptide bonds are bonds between 2 amino acids. They occur between a carboxyl and an amino group. They're a type of covalent bond where the carbon and nitrogen are sharing electrons. And this can happen over and over and over and over again to start to build a long chain, right? So the basic unit of a protein is an amino acid. Amino acids have certain parts that are the same and certain parts that are different or unique. So the unique parts of these R groups, and again, at this point, they're not coming into play. They're just there. They come into play later on in the structure. As we start to build a few amino acids joined together, so a couple of them like this, could just be referred to as a peptide. So a peptide may be sort of a short group of amino acids. There's a lot of places in cells where peptides, just a couple, like 4, that's it. Amino acids, they're signal molecules, they help communicate information in cells, and they have a variety of other jobs. So short little peptides, they're not a single amino acid, but they're not a giant big protein either. They're just something in between, and they're not called proteins, they're called peptides, and that's because they don't have complicated folding, which is what we'll get into with the proteins, right? But they do have amino acids joined together with peptide bonds, and that's kind of a peptide. Polypeptide is just when it just starts to get bigger and bigger and bigger. I mean, it could be really big. It could be full size, the full size of an actual protein, and it can have a lot of complicated folding to it as well, but it doesn't have function. So that's the next thing we're going to look at here, is the difference between these two terms, protein versus polypeptide. What's the difference? Big structural molecule, this is pretty much the same molecule, but now it has function. So many of these molecules will have something like a molecular on-off switch. If they're shaped exactly the right way, then they start to function, and without that shape, then they won't function. They'll exist, they're there, all the parts are attached, but there's one little bit more that has to happen, and if it doesn't happen, then they don't work. It could be related to pH. So for example, the cells that line your stomach, the cells that make your stomach lining, they make the enzymes that digest your food. So they break down carbohydrates and lipids and proteins. The cells are made of carbohydrates and lipids and proteins. So how is it that the cell who makes those enzymes isn't destroyed by those enzymes? And that's because when they're made, they're at a certain pH. They're at pH 7, say, for example. And at pH 7, those proteins, or technically they're polypeptides at this point, they're not folded properly, so they don't actually work. They don't do their job. But when they leave the cells and go into your stomach, into that compartment, it's very acidic in there. And so they refold and change their shape, and then they become active proteins as enzymes who carry out digestive activity. Okay, so that's just an example. Something's sometimes a phosphate group, like I mentioned in one of the others, that sometimes is a molecular on-off signal. So phosphates sometimes join to things, and then they turn them on or turn them off. Usually turn on when they're attached. But there's things like that where some group gets attached, and that's what turns on or activates a protein, or activates a polypeptide, giving it function, so it's now a protein. So it's a very, very slight different term. Some people use them interchangeably, and in many ways they are interchangeable. It's just there's a fine difference between polypeptide having function, carrying out job, and then the way the word is used when we carry out the jobs, they're done by proteins. So it's very slight, but it exists. So as we start to build these structures I keep talking about, we have something called primary, that's the sequence of a polypeptide, and that is just the amino acids joined together. And that's what I'm doing here. Join amino acids. I'm building primary structure. There's something called secondary structure. So we're talking here about structure, okay? Secondary structure is a little bit of folding, right? And there's two main types. There's secondary structure where these things then start to get twisted into a helix, like that. Or alternatively, instead of a helix, they get shaped into this zigzag, sort of back and forth shape like that, which sometimes then gets a third shape, which is just like a random loop like this. And then they can kind of go back and forth, and then they do this sort of thing. So this one is called an alpha helix. This little symbol is alpha. And this one's called beta, that's a B, a Greek beta sheet, where there are many of these kind of joined together. And so that's the secondary structure. There's called tertiary, and that's three-dimensional folding. And the key thing I want to point out about the tertiary structure is this is where the R groups come in. So it's these R groups, that's what determines the three-dimensional folding a lot of times. So positively charged ones and negatively charged ones attract each other to, say, form hydrogen bonds. Ionized ones might attract each other. Non-polar ones might be pushed together. Polar ones might, attract each other. So there's all different sorts of ways that the R groups then come in later on, but not really until the tertiary structure level. At this level, they may become, once they do this folding, functional. Got the squeak. Here, there we go. They may become functional. So so primary, they're not going to be functional unless they're just a peptide that carries like a signal, like I mentioned. So it could be a few of these. That's all they are is primary structure, and they do technically have a job, but they're still referred to as a peptide because they're tiny. As they get bigger, they're just referred to as polypeptides structurally, and they don't really have a job yet. So they're being folded, and they can have these levels of folding, twists and coils and zigzags. Again, they don't quite have their function yet. They need to be folded in a much more three-dimensional complex way, and that shape gives them their function. And then they can carry out their job. Some of them don't do that. They're still a fourth level. So they're still what's called quaternary. So this is the level 1, level 2, level 3. And level 4 is called quaternary. And that's where we have multiple polypeptides bonding. So that means you have one polypeptide, but it doesn't carry out any job. It's got all the stuff, it's got all the folding, but it doesn't really work. And then you have a second one, totally independent. It goes through all this. Built, folded, doesn't have a job. But when the two of them link up together, then they actually work with each other, and that the protein is actually the two of them combined. And that's again why I really emphasize the difference in the terminology, because many times a protein, a functional protein, is a combination of multiple polypeptides. And that's where we get this quaternary structure. So say an example of that is your hemoglobin is an example where there's four different polypeptides come together. If you take each one individually, they're not going to carry oxygen. But you need all four separate protein or polypeptides here to come together to form this protein called hemoglobin that carries oxygen for your body. Tons of details, more that we could get into, but really for your class, just being emphasized. Proteins are very important. Their job in the cell is going to come up. Most everything cellular that we're going to talk about is all going to be related to proteins. So they're going to be enzymes that are all about proteins. Even the DNA replication is about proteins doing their jobs. The gene expression is about how to make proteins. So really a lot of this next material is all going to be about them. So this isn't really the end of them. This is just more the introduction to their terminology and some of the basics of their structure. So we're not doing nucleic acids until later. So the next thing we'll actually go into as a topic is the beginning of cell membranes. So we'll start to actually talk about, but done with the chemistry part, at least alone, and now we're going to be talking about the cells, and there'll still be chemistry, but it's all related to cell structure.